Hi everyone, welcome back. So previously we talked a lot about cell membranes and walls for bacterial cells. We explored cell membranes as this barrier and container for the cell and as this really important tool for generating ATP for those cells. And then cell walls being this very important structure that gives rigid protective support and also exhibits some significant variation from one group to another between gram-positive and gram-negative varieties. Now, that's not the only thing that is very distinctive about bacterial cells. So we're going to talk more about some important features of those cells here. So outside of the cell wall, we can sometimes find a structure called the capsule, otherwise known as the glycocalyx, which is what I tend to say. Glyco meaning sugar, calyx meaning coat. This is really a slimy covering that kind of encapsulates the cell. If it's present, it'll be outside of the cell wall, and it's mainly made of sugar chains. And it's got this really interesting quality that essentially turns it into a bit of a jelly-like structure, very gel-like. And this structure can serve a bunch of different functions. First, it can help the cell attach to surfaces. It can also be a barrier that protects it from threats, like from viruses that would try to attach to and infect the cell, from our immune defenses if it was a pathogen, even other bacteria in some cases. It also lets the bacteria help retain moisture because that even when it dries out, even when the environment dries out, the glycocalyx can still retain some moisture for at least a little bit. And it also helps with absorbing nutrients from the environment. So, this is a structure that can be very helpful. That said, not all bacteria actually have a glycocalyx. So it is variable. We can also find flagella on cells. Not all cells actually have flagella, but of those that do, this is how they get around. A flagellum, this is a whip-like structure that can move around and essentially pull or push a cell in specific directions. Now this is it's made of a bunch of different protein filaments that are all kind of aligned in specific ways and work. They're like pulled together to allow this cell to move. And some bacteria have them, some do not. And so the ones that do are motile, as in they're capable of movement, and the ones that don't are non-motile, incapable of movement. Among the ones that do have flagella, they could have an incredible wide variety of arrangements. So some species have like what we call peritrichous flagella. E. coli is a great example of that. These are essentially they're covered all around their body in flagella, and that means they can pull or push their body in a variety of different directions. You could have clusters of flagella on one end or the other end of the cell, and so that can create like a pull area where they can thrash their flagella and push themselves in specific directions. And then in some cases you could only have one flagellum, where we have just one at one end being a polar arrangement like this. So incredible variety of how this can look in bacterial cells. So bacterial flagella, the way they work is pretty interesting too, because previously we talked about the way that cells can generate ATP through this process of moving around hydrogen ions and kind of making them flow like water over a water wheel. So that structure is how we use to generate ATP. It's how our mitochondria generate ATP, and it's also how bacterial cells do this on their cell membranes. But the really interesting thing is this is also the same mechanism that bacterial flagella are powered by. It's the same basic concept. We have a rotating wheel that spins through the power of moving hydrogen ions. So these cells kind of do double duty when they make ATP. They also can move ions that can allow them to move as well, that can actually like turn their flagella, which is really interesting. It's sort of like a convergence on strategy there. We can also find some additional features on the outside of cells. We can find pili and fimbriae, okay? So these are two projections, or like little proteins that can arise from the cell surface, okay? Pili tend to be longer than fimbriae. Both of them are useful for similar things, for both like surface adhesion. Essentially, it's just more surface area to grab onto a surface with, and also sometimes for very simple movement. Allows the cell to kind of do a bit of a twitch or a jitter as opposed to a full forward movement. Inside the cell, we could have a lot of different things. One of the first things we're going to talk about is inclusion bodies. So these are essentially they're vesicles. They're little inner membranes that can form, and basically their only job is to contain stores of different materials. And so not all species will make these vesicles. Some will not. They serve a bunch of different functions. Okay, these inclusion bodies are mainly granules. We call them granules, meaning that they're solid materials. They contain like crystalline things. So one of those things is a starch or glycogen store. So essentially, this is like how these cells might be temporarily storing energy to use for later. Some species can store sulfur in different forms, which can be used for energy production, again, for specific species that can use it. Sometimes species can store magnetic particles, which they use for magnetoreception. It's basically like they can detect like the poles of the Earth and things like that, and orient themselves in specific ways. And then in other cases, these inclusion bodies can be gaseous. So they can contain like stored gases they can use to maintain buoyancy. So they can float or sink depending on how much gas they have. And essentially using that, they can figure out where they would like to exist in the water column, like in a fluid environment. This is kind of what they can look like. There's a variety of different forms that they can take. But this is one example of a species that can actually like synthesize and store like PHBs, which is like really it's kind of cool because we can use those to make biodegradable plastics. Now let's talk about the genome of these organisms. So prokaryotes have their genetic material stored pretty differently than the way that we do. We store our genetic information inside of our nucleus. We have a dedicated organelle that does this. But again, prokaryotes don't have organelles at all. So what they do have sometimes, what they do have, all the time, is a nucleoid region. Okay, this is the central space where DNA is stored. It's not bound by any like membrane or anything like that. It's called a nucleoid because it's nucleus-like. It's very distinct from the nucleus, but it's similar in the sense that this is where DNA is stored. So DNA is kept here and maintained here by a variety of proteins that help keep their DNA healthy and organized. And this is also where DNA is read, where it is used to make proteins. Now, most eukaryotic genomes are divided up across multiple chromosomes. Okay, so a chromosome is just a large region of DNA, usually a very distinct chunk of that DNA. Now, humans, being eukaryotes as we are, we have many chromosomes, and we have 23 pairs of chromosomes. So 2 pairs of 23, 46 in total. One that we got from one parent, one that we got from the other parent. And our chromosomes are also linearly arranged, where if you imagine a chromosome kind of like a very, very long rope, there is one end to the rope, and as you go to the other end, you'll reach the end. There's a start and end to this set of DNA. Prokaryotes are very different, though, because they only have one chromosome. Usually, they only have one copy as well, unlike us, although there are examples where that can change. But that's a pretty major thing. They only have one chromosome. They also have this chromosome arranged in a specific way where it's a continuous loop. We call it circular as opposed to linear, because those ends of the rope are tied together. They meet at a central point. Okay, so there really is no start and end for these genomes. So there's actually a lot of similarity that these bacterial cells share to mitochondria, because eukaryotic mitochondria are really weird. They have their own genome. It's separate from the nuclear genome that we have in our nucleus. And it's circular, actually, very much like bacterial genomes. And curiously, mitochondria also have a second membrane, one of them inside the other. It's very similar to some things that we see with some bacteria. And so we have this running question that researchers actually asked for quite some time, which is, after we had learned about it, why? Why is the mitochondrion so similar to bacteria? Well, our current theory is that mitochondria are descended from bacteria. That this is a really interesting piece of history that we believe that we've really worked out here. That way back in time, go back a billion and a half years, right? The early eukaryotes and the early prokaryotes had recently diverged. And these eukaryotes, who have a primitive nucleus and some simple organelles, swallowed a bacterial cell, this proto-bacterial cell. And, as it probably usually did because it needed to eat. Except that instead of digesting that cell, it actually kept it around. It didn't manage to eat it, and it survived inside of the cell. Kind of sitting there and standing there for a long, long time, existing, replicating. They probably stole resources from each other. Eventually, they began to change. They refined. They simplified. Because if they started a positive relationship with one another, where they weren't stealing from each other and instead they were sharing, they didn't really need to do all the same things. The bacterial cell, this protobacterial cell, it didn't have to worry about acquiring resources anymore because the host cell would provide it. It didn't have to worry about moving because that was something that the host cell did. It didn't have to worry about a bunch of different things. And so the genes that regulated that process could undergo mutations and essentially be lost. Similarly, the host cell, this eukaryote, it suddenly had an ATP factory living inside of it, and it was sharing ATP as a resource. And so making ATP wasn't something that the eukaryotic cell had to worry about as much anymore, and so it could lose those genes as well. Eventually, these things simplified so much that the mitochondrial this now organelle, is completely dependent on its host. And it also only has genes that are related to its function, to its core function, and that is making ATP and also replicating itself. That's it. And so this is a pretty substantial change that has undergone where this species, these two species formed this ancient symbiosis and then slowly became impossibly dependent on each other to the point where they could not be separated without killing one another. This is something that we call this endosymbiosis. Okay, endosymbiosis, where it's a symbiosis where one organism lives inside of another. And so what we saw was that the ancient mitochondria and its host cell, it divided roles between them. The ancestral eukaryotic cell mainly focused on homeostasis, keeping those healthy functional conditions going in the cell, collecting nutrients and energy sources and offering protection for that proto mitochondrion. And then the ancestral mitochondrion, had its own role, and that was making ATP. It was all about ATP production, and it just got really, really, really good at that. Okay? A similar process probably occurred with chloroplasts, actually, for plants, because we have very similar situation where chloroplasts have their own genome, and they look very much like bacterial cells. They have characteristics of cyanobacteria. And so we think that's probably what happened to them as well. A very similar history of acquisition. So, enough about mitochondria. In addition to the chromosome that these prokaryotic cells have, bacteria also frequently have plasmids. These are very small circular bundles of genes that are separate from the nucleoid genome. And cells could have none of them. They could have one or two or hundreds in some cases. And they are often very useful to the cell because they can contain a variety of different genes that can help them survive in otherwise adverse conditions, like antibiotic resistance or the ability to produce a specific toxin or unique metabolic abilities, for example, like allowing it to digest something it didn't used to be able to use before, things like that. So this is kind of what that looks like. We have a distinct separation between bacterial DNA, like the genome itself, and the plasmids that it may have. And those plasmids can actually come and go. They can acquire new ones and in very, very strange ways that we'll definitely explore later. So plasmids are really small. I guess we'll explore it now. Plasmids are really small. It makes them really easy to move. And that is pretty useful to bacteria because they can actually pass them between one another. We have a secondary structure that we see sometimes with bacteria called the sex pilus. It's a distinct version of the pili that is long and hollow. Essentially, it is a plasmid transfer tube. That's what the sex pilus is. It allows these bacteria to actually trade with one another. If they sidle up next to each other and recognize that one of them has a plasmid that they could trade, they can make a copy of it and pass it between cells, which is really interesting. Now, unlike humans, which can really only mainly pass genes from parent to child, something we call vertical transmission, bacteria can trade genetics with their neighbors, actually. They can pass in this very unusual way, with existing cells. So they can just go right up next to one another and then pass back and forth. This is something that we call horizontal gene transfer, or HGT. Okay, this is very different from vertical gene transfer. That's what I was talking about before, like what humans do when you pass your genetics on to your children or you inherited your genetics from your parents. That was vertical, basically moving down in generations. But horizontal gene transfer is basically any other kind of transfer. When you move genetic information between individuals that currently exist. So two neighbors bump up next to each other and then pass DNA. They become hybrids of one another. They suddenly contain DNA that one another had, that was once just in them, but is now in both of them. Sometimes this can be an intentional thing where the bacterium is making a choice to do this, where it actually, in the case of conjugation, which is what I was mentioning earlier with the sex pilus, they intentionally make copies of their plasmids and pass them between one another. Sometimes it's also unintentional. In the case of a virus accidentally inserting foreign DNA into a genome as it tries to infect the cell. Bacteria utilize these strategies pretty often, mainly because they don't reproduce sexually, and so they don't have a partner. They don't really have an easy way of getting new genetic information, other than mutation. Mutation just randomly, and mutation is random. It is a random change to your DNA, so it could be quite bad. It's very possible to get a mutation and have it be incredibly detrimental and damage some core function that a cell has. Horizontal gene transfer allows cells to be actually a bit picky about what they have. And they can pick up DNA from different places, and if it's useful, they can use it. They can integrate it into their cells. This is really notable for bacteria, and it's a very important way that they actually acquire new genetic information, particularly important in medicine when we're talking about bacteria that can be, acquiring antibiotic resistance or virulence factors that allow them to be more detrimental as a pathogen, for example.